Hello, everyone. We're going to be starting here in just a few more minutes, so please be patient. And we will be also streaming this webinar on YouTube and recording it for others who are not able to join because of the time issue. So as we start, please keep your microphones unmuted, and we'll be starting here in just a few more minutes. Again, just a reminder, we're going to be starting here in just a couple more minutes. So thank you for your patience again, and we're looking forward to this webinar together with you. Okay, hello and welcome to this webinar. This webinar is dedicated to our upcoming program called Omics Logic Bioinformatics for Precision Oncology. So you might be asking yourself, what is Omics Logic? And to tell you about it, I'm going to just share a few brief points that are a central component of our vision. So the goal of Omics Logic programs is to provide training in bioinformatics, enabling independent research guided by mentors and peer example. It is also to develop a community with shared passion for data-driven research and an appreciation of citizen science. We also seek to enable students, clinicians, and faculty of all backgrounds to develop novel and independent research using latest technology in life and data sciences. And so over the course of the past several years, our team has been collaborating with a number of academic institutions around the world that have participated in content evaluation, curriculum review, and project design. And so as a result of this, we believe that the Omics Logic curriculum and programs have been thoroughly evaluated, and now we have evidence that they lead to excellent outcomes. So as you have joined this program and this webinar, we ask that you also join us on social media. So we have these two groups, one on LinkedIn and one on Facebook. I think what's interesting about this is that we have been able to attract a very high caliber of people, both from research, industry, and different academic institution environments. And so we are looking forward to interacting with you, sharing recent updates from different research articles related to different topics in bioinformatics, and also learning more about your interests. So what we will discuss today and what we prepared for you and throughout this program is the result of hard work by an amazing team led by company co-founders, Dr. Alfred Tauber and Dr. Leonid Brodsky. Dr. Alfred Tauber comes from a medical background and has extensive expertise in oncology, immunology, biochemistry of inflammation, and has been recognized internationally for his generous support of many research projects and initiatives that support data science training and applications in medicine. And Dr. Leonid Brodsky is currently the director of the Tauber Bioinformatics Research Center at University of Haifa and is an expert in bioinformatics and biostatistics. He has a background in mathematics and has numerous publications in mathematics and bioinformatics, and over the course of his career has developed multiple novel algorithmic approaches to biomedical data analysis. The rest of the team includes experts in bioinformatics, computer science, and training. So some of the people that you see here will also be presenting today. Now, this program was designed to address some of the challenges that are common to research in oncology, where omics data is becoming central to the research that drives novel diagnostic approaches, as well as therapeutic insights into how cancer can be treated better. So first of all, I wanted to share with you a little bit about why cancer. So cancer is one of the leading causes of morbidity and mortality worldwide. In fact, it is the second cause of cancer after cardiovascular diseases. But as you can see on this map, it is a problem of the Western world. And there are several reasons for that, but we might already consider how it's related to lifestyle, the length of life, so aging, and also consider the different types of approaches to treatment and prevention of cancer as well. And so because cancer is primarily a disease that is manifested at the cellular level, we understand this as a progression from healthy cells to abnormal cells to then cancer that is contained or pre-invasive. And then eventually when the cancer becomes to spread throughout different tissues, that is typically when it is detected and then treated. And so part of what we are going to be speaking today is the research that is enabling us to screen for and identify cancer early on, also developing techniques and novel insights into the molecular mechanisms of cancer that would allow for better treatment of cancer overall. So one of the things that characterizes cancer research, both basic and translational oncology research, is the collection of high throughput molecular data that could describe different types of mechanisms that regulate gene expression and the regulation of subcellular processes that might lead to cancer development and progression. And to study these mechanisms, not everything is possible to do with patient data. So a lot of it has to do with animal models, as well as cell line culture and organoids that allow us to study many different perturbations at the subcellular level. And so as a result, we are seeing this increase in big data that comes from next generation sequencing and other types of omics data that together can help us find answers to this important condition. Now, one of the things that we will want to do in this program is provide you with an opportunity to gain insight into how to analyze such data, identifying variation at the genomic and transcriptomic levels, and understanding both the causes of cancer, the hallmarks of cancer, and some of the ways that this disease progresses into different stages. So one of the important types of data that we will speak about is next generation sequencing. And one of the primary ways that you can analyze next generation sequencing data in the context of oncology is called variant calling, where we can align multiple reads to the reference genome, identify germline mutations, and then to also see somatic mutations that might occur as a result of cell division. Now, ultimately, those mutations lead to changes at the mRNA and protein level. And so we will also take a look at how to process and analyze next generation sequencing for gene expression analysis. So we'll take a look at different steps of mapping, quantification, and differential gene expression that would allow us to compare a given sample to a control and really understand different types of conditions. Now, oftentimes people think about the analysis itself as a way to gain insight into the biological components that are manifested in pathways and gene ontology terms. And so we will talk about how to use the same user-friendly tools to understand gene set and pathway enrichment analysis. Ultimately, these types of research data sets are allowing us to see the basic mechanisms that define cancer. And so a lot of times, because of the limitations of these cell lines, organoids, and animal models, we would also like to understand the real patient situation and kind of what happens in real life. And so because of that, we're going to be looking at such data repositories like the National Center for Biotechnology Information, or NCBI, as well as the Cancer Genome Atlas that contains a number of important data sets, multi-omics data sets, from real patients with a lot of clinical information. Now, we will be able to see how real collections of data can show us whether the insights from different types of cell line studies and animal model studies could really be applicable to the patient population at large. Now, we will also take a look at some interesting publications that refine our understanding also about the effectiveness of treatment of different types of cancers. And so there are collections of cell line high throughput experiments that essentially are designed to illustrate the response of different types of cell lines to different types of treatments. And because these are cell lines derived from real cancer patients, they provide a good insight into a predictive analysis of what might work for a given situation. Now, when we turn to the TCGA data, we will see how there are a numerous collection of risk factors, outcomes, all kinds of metadata that can inform these decisions about treatment of cancer. And so we will review this literature, understand what are these different types of factors that should be considered, and learn to incorporate them into our analysis. Now, to do all of this in a short period of time, we will need some effective resources for learning and research. And so what we've designed in the Omics Logic Bioinformatics training programs are important resources that will help capture this important information about the biology and the background of cancer, the practical bioinformatics analysis methods, as well as translational skills that you can learn and apply to a number of different projects that you might be interested in. So for the different sections of this program, we will leverage a number of different resources that you can benefit from and use to learn about genomics. For example, asynchronous coursework with theoretical materials, quizzes, and practical assignments, cloud-based analytical server where you can analyze large genomic data and visualize the results of your analysis, the Omics Logic Playground where you can practice and learn coding in R and Python, as well as a repository of curated project examples with data sets that you can try and analyze and get reproducible results. Now, the courses and projects that we have prepared for this program include training as an introduction to bioinformatics methods, an introduction to genomic data, transcriptomic data, and the application of bioinformatics in the context of several different projects, including early detection of cancer, an approach to personalized precision treatment of cancer, as well as the immune response of different cancer factors. Throughout the course, you will also be given directions to access the T-Bioinfo platform for bioinformatic processing and analysis of genomic and transcriptomic data. The platform includes demo pipelines, as well as a data management console and analysis cloud infrastructure that you can run bioinformatics pipelines for large data sets. Different stages of analysis are performed in different sections of this multi-omics platform. In many cases, you will also be able to take elements of that pipeline and learn how to perform the analysis using R and Python. But before we continue to speak about all of these different resources and the program itself, I wanted to launch a poll really quickly and get your feedback on a few different questions. So the first question that I have is, what is your current country of residence? So we have a few different options: USA, Canada, Mexico, Brazil, Peru, Argentina, Ecuador, or other. The second question is, what is your current level of education? So it could be high school, bachelor's degree, master's degree, PhD, faculty or postdoc, or other. And then what is your current area of interest or specialization? Bioinformatics, oncology, undecided, or other. So let's take a couple of minutes to answer some of these questions, and then I will pass this presentation over to Clinton, who will tell us more about his experience with cancer research and some of the program resources that we will be going through in this bioinformatics training program.
All right.
Okay, so Clinton, I'll pass it on to you. You can start sharing. Okay, thank you.
All right.
And
So
I'll keep the poll on the screen, but you can start your part of the presentation.
all right, that sounds great, Ilya. Thank you. Hello and welcome everyone to Bioinformatics for Precision Oncology. This is a 1-month program that will teach you the foundational skills to analyze complex cancer data sets in hopes to achieve a scientific question. These skills are transferable to other types of data sets. However, the primary focus will be oncology. In my part of the presentation, I will speak about my personal experience with bioinformatics and how it led me to conduct research studies that looked at the basic biology behind cancer. I will review the program schedule and explain how the different topics fit together, providing an experience for students and researchers that are interested to apply bioinformatics to research studies related to cancer. I will also discuss some ways this program can benefit you, such as the development of practical skills, critical thinking, and research. Finally, I will talk about what you can do to get the most out of the Omics Logic Bioinformatics for Precision Oncology program. Precision medicine is changing the way we understand, diagnose, and treat cancer. This transformation is driven by high throughput molecular data from patients, animal models, and large-scale cell line experiments. What I will share with you today will introduce you to the Omics Logic Bioinformatics for Precision Oncology program, which is designed to provide training and guidance to students and researchers interested in various omics data. But before we speak about the program, let me share a few points about my background and the reason why bioinformatics is central to translational research in oncology. My name is Clinton Kuna. I completed my bachelor's degree at Carnegie Mellon University, where I discovered my passion for research. Later on, I decided to continue learning about life sciences. As a result, I now have 2 master's degrees, one from the University of Guelph and the other from the University of Ottawa. Since I was interested in translational bioinformatics, I did a master's in bioinformatics and another one in molecular medicine. As a result, I am familiar with biomedical research and as well as data analytics. Cancer is extremely heterogeneous. Even though the war on cancer that was announced 70 years ago has brought about many advances in our understanding of the basic mechanisms that lead to tumor growth and metastasis, it is also clear that current research did not solve this problem. On the contrary, we are learning more and more about this complex condition. Now, to show you how complex cancer is, let me give you an example from my past research. There are several proteins that play different roles within certain contexts. For example, ST20-like kinase, or SLK. SLK is a protein that plays an important role in cellular processes, such as proliferation, migration, and apoptosis. It affects embryo development and is shown to play a role in tumor genesis. This was studied in a new induced mammary tumor model in mice. These experiments showed that SLK knockout mice developed tumors faster and showed an increased expression of SOX10, an important biomarker in cancer. However, in my own research, we studied SLK in mouse embryonic fibroblasts, where SLK knockout reduces focal adhesion turnover. It turns out that SLK plays different roles depending on the context. Even though this contradicts the other study, it also shows how complex the biology of cancer can be and how careful we have to be with experimental design and data analysis to truly understand its pathogenesis. Research on SLK is ongoing. This is similar to cancer. Every cancer is different, and it's a challenge to determine why a knockout reduces a process in one context, but increases it in another. My master's thesis gave me a glimpse of how bioinformatics can be used to study oncology, and the skills I acquired enabled me to learn more or use more sophisticated methods of data analysis, such as machine learning and other types of algorithms. So there's a lot of complexity with high throughput data, and that you need complex machine learning algorithms to deal with it. Therefore, I decided to pursue a master's in bioinformatics and learned those complex skills. Pure biology is good, but having the data analysis skills to create meaningful relationships definitely solves major problems that has been translated into applications within medicine. Bioinformatics has exposed me to other fields due to its interdisciplinary nature, such as statistics, engineering, and mathematics. Does anyone have any questions related to the transition from biology to bioinformatics? I have been through it, and you can too. Are most of you biologists, or do you have a bioinformatics background?
I'm a biologist, and I'm doing some bioinformatics in
Nice.
my lab. Yeah.
Nice. You can, if you feel shy to use the microphone, you can type in the chat.
Okay.
Yeah. All right. This program is mainly for biologists, citizen scientists, and people who have a genuine interest in cancer and biology. So it's a great foundation to get into the field of bioinformatics, especially if you have no previous knowledge, because we have the toolkit to help you get up to speed. So now let me explain how anyone, including you, can learn the same skills as I have and apply them to a data set by completing this program. The Bioinformatics for Precision Oncology program starts on March 15th and will go through April 9th. During this time, we will learn to apply analysis of genomic and transcriptomic data to the study of cancer biology. Our focus will be on bioinformatics methods, but we will use examples of data sets taken from oncology projects. At the end of the program, we are encouraging you to apply the learned skills to data sets of your own choice. During that time, we will support your research to make sure you can process, analyze, and interpret data. We will learn important concepts within cancer biology, such as the central dogma, the hallmarks of cancer, and how cancer is diagnosed and treated. Several bioinformatics methods will be talked about, such as processing, variant calling, RNA-seq, and more. Finally, we will look at bioinformatics method. We will look at sample projects and learn how to use our own projects with our newly learned bioinformatics methods. By learning these major concepts, you will be well equipped to start your own projects. So if you haven't checked out the Precision Oncology link, here is a snapshot of all the topics we will cover. You can register with the link above, and I will re-show you the link at the end of the presentation. In the first session on March 15th, we will review introductory material about oncology and the challenges associated with big data in cancer research. In the session after that, we will start talking about next generation sequencing and what this data can reveal about biological processes and the clinical manifestation of cancer drivers, tumor development, and the tumor microenvironment. Finally, in the last session of the first week, we will speak about challenges of big data sets that have to be processed, analyzed, and annotated to yield insights into cancer biology and discuss what types of information we can extract from next generation sequencing data. In the first session, we will start talking about cancer biology and the cellular and tissue levels, learning about the role of DNA changes and gene expression variation related to cancer risk, progression and its outcomes. In order to conduct research on cancer, one needs to know its fundamentals. The content of this session is related to what would be taught in the beginning of any cancer-based program, such as a master's degree. This content can equip someone who is completely new to the field of oncology, know foundational concepts for more advanced studies. One of the first concepts that we will learn would be the hallmarks of cancer. What defines a cancer? These are distinctive and complementary capabilities that enable tumor growth and metastasis. These hallmarks have evolved over the past decade. The main concepts of the hallmarks involve rapid growth, rapid spread, evasion of the host cell's defenses, increased mutation rate that transfers to its daughter cells, and finally, immortality. We will speak about the challenges of big data sets that have to be processed, analyzed, and annotated to yield insights into cancer biology. Topics that will be covered include hypothesis testing, data mining, statistics, and how to use the T-Bio platform. Has anyone taken a statistics course? And what are some techniques that you've learned or are familiar with? Please write a word or a sentence, a one sentence in the chat, just to get an idea of your statistics background. Yes. Okay, perfect. taking stats. Are you an undergraduate, Sylvia, or a graduate? What's your level of education slash expertise? Doctor, nice. Two-tailed, yeah, nice, nice. So great. You'll learn how to use similar techniques within this program and apply them on oncology data sets. The growth in high throughput sequencing of cancer genomes and transcriptomes has produced a lot of data, which slows down the progress of cancer biologists and oncologists from quickly and efficiently gleaning knowledge. There are so many data sets that need to be analyzed in order to find relationships between cancerous processes, tumor genomic profiles, and treatment response. That's why we will learn about these data types and how to efficiently process and analyze such data. So just what are the largest data sets that you've all worked with? You can type a gigabyte amount in the chat or roughly describe the size. What's the largest data set you've ever worked with? If you guys can remember that number. I'll give you mine. This is just, I worked with something that, nice. You can write your Excel sheets as well if you haven't worked with larger data sets. Maybe I think Excel, the maximum might be around 250. I don't know if you guys have ever probed into the size of your data sets. But it's fine. You will learn how to analyze and work with big data sets in this program. Nice. Wow, 20 gig. That's pretty big. Nice. So you will learn how to analyze extremely large biological data sets using a remote system. The T-Bio platform has a large storage space and it can handle large projects. So you can download really large data sets and work with them remotely. So can everyone, you don't have to write a lot. You can give me maybe a sentence or a word. What's your field or experience in the field of bioinformatics? You can say none if it's your first time, or you can give a one sentence description. If you've, okay, R, nice, yes. That's really good. Anyone else? Has anyone never really used bioinformatics? You can say none. None, okay. Yep. Well, this is actually a great starting point. So if this is the first time you've heard of Pine Biotech, Omics Logic, or even bioinformatics, I highly recommend checking out the following link on the screen. Please start going through the Introduction to Bioinformatics course we have on the following web address. If maybe Ilya could paste it in the chat, or I can paste it at the end of the, all right, perfect. Yeah, this will give you a introduction to what bioinformatics is, define the field, so you can have that foundational knowledge. Nice. Wow. 15 separate subjects. Wow, that's great. That's great. That's great. I can see you potentially upgrading your work to maybe a graduate degree at some point. So we will also discuss the types of information that can be extracted from next generation sequencing data. You will learn about how to process, analyze, and interpret genomics data through specific pipelines. Example techniques include variant calling, mapping, and finding the significance of specific mutations. These are common techniques used in hospitals to analyze sequencing data from cancer patients in hopes to diagnose and potentially design an effective treatment plan. With this skill set and a few projects worth of experience, you could potentially be on the path to help a hospital near you. So, can someone tell me the purpose of looking at DNA variation? There could be multiple purposes, but maybe in a sentence or one word, how would you describe, why would you want to look at DNA variation? Like, what purpose does that serve? Why do I want to look at DNA variation? Okay, I guess, perfect. Okay, epidemiology of the virus. Yeah. All right. So, if you'd like a more general, yes, okay, to see how cancerous cells operate for different types of DNA in people. Yeah, those are nice answers. If you'd like a more general and in-depth answer, please check out the courses on the link on the screen shown here. Check out Introduction to Genomics and Genomics 1 courses. They focus on answering the following questions. What is DNA variation and what types of variation are there? What DNA elements are being studied for variation? And what types of genomic information exist and can be analyzed? Yes, it could explain some changes in the phenotype, exactly. So if you, yeah, within the precision oncology program, you will learn how to load NGS data and construct data analysis pipelines to find genetic variants in breast cancer. And you also do similar tasks in the genomics program. So most of you come from a biology background, so this question might be easy for you. What are 3 common types of DNA mutations? Yeah, okay, yeah, DNA expression. That could also be another reason why we look at DNA variation. Okay, missense. Okay, that's good. Nonsense. Yeah. Okay. There are more than 3, but you can give me 3. So you gave me 2, which is fine. So yeah, deletion. Yep. So yeah, insertions. Perfect. Yes. So what some of the common ones are insertions, deletions, substitutions, and structural ones. So you might have an extra chromosome. And usually in cancer, these mutations affect the following. So they affect tumor suppressor genes, oncogenes, or DNA repair genes. So that once those are affected, then the system, it starts to get buggy, and then the hallmarks of cancer come in, and then the cancer forms. So the images shown, the images above show different single nucleotide polymorphisms and some of the steps in the NGS variant calling pipeline. So variant calling is useful to determine which changes might cause a particular phenotype, just like who said that? I think someone mentioned the phenotype. Yeah, Matthias. And there are 3 types of mutations, just like we observed before. And during this program, we will map, visualize, and detect significant changes in DNA sequences using a reference genome and paired tumor normal samples. We will also spend considerable time learning about gene expression and RNA-seq, practicing each of the analysis steps on the user-friendly T-Bio platform. You will learn how to process, analyze, and interpret next-generation sequencing data. So for a more in-depth course that focuses just on transcriptomics, check out our Transcriptomics series. These courses provide a broad but detailed overview on key steps such as processing, statistical analysis, machine learning, and single-cell data analysis techniques. So they cover various methods, various terms, provide practical examples, and they also have quizzes to check your understanding. The precision oncology program will also use key concepts from these series, but it's more geared towards cancer data sets. So has anyone heard of the term differential gene expression? You can either say yes or describe it in one sentence, whichever you feel comfortable with. So have you heard of differential gene expression? Okay, yes, perfect. Oh, we have a lot of biologists here. I should see like several yeses. Yes. We can relate two samples, yes, and it depending on it's checking the significance of genes between two groups. So I'd say it's more of two different groups versus two completely different diseases. But I think there's a specific pipeline for that. But within the context of this course, we're going to focus on two different groups. So maybe one group one's the normal group and one's the disease group. So related diseases, I believe we can, but we won't be doing the example within this course. Okay, perfect. So researchers need a way to assess whether gene expression values have an actual significance between them, or whether those expression values were caused by a technical issue. So differential expression analysis means taking normalized gene expression data and performing a test to see if there's significant changes between experimental groups. So one example is that we will prepare and run a full processing pipeline designed for differential gene expression. So here's an example. So here's one condition and here's another condition. So one might be normal and one might be disease. So we will also learn to apply statistical approaches like regression to study differences between conditions that have multiple factors. Other techniques include PCA, t-tests, and differential gene expression. So factor analysis models are used to describe a large number of observed variables by a smaller number of unobserved variables, which are known as the factors. These factors explain all correlation between observed variables. So by doing a factor regression analysis, we will find relationships in data and find significant genes that are associated with cancer types or stages that aren't noticeable at first. So essentially what we're doing is finding hidden variables that can help us find stronger relationships for future analysis. So I'll give you an example to explain this concept. So for example, suppose there are 20 movie reviewers and they're asked to rate a movie from 1 being bad to 5 being excellent. to different types of movies. The first movie is the funny movie. The second one is This Will Make You Laugh. The third one is Forbidden Planet. Fourth one's Going to Mars. Fifth one's The History of China. And the sixth one is How Is the World Changing? And the last one is How Were the Pyramids Built? It's possible that the reviewers assigned ratings that may be correlated with hidden variables, such as comedy, science fiction, and history, because one movie was called the funny movie and the other one was called This Will Make You Laugh. And there may be the variable hidden, or the variable comedy is a hidden variable. Based on the previous session's analysis, we will build upon that foundation to deduce which genes are important for future use in biological interpretation, using several methods that will help us gain a systematic understanding of the found genes. Without this step, we would just limit ourselves with understanding that these two genes are significantly different, but we wouldn't be able to translate that to a observable phenotype. Most companies seek bioinformaticians to act as a bridge to annotate and interpret their data for an audience who might not understand the scientific complexities. What are some genes that you know that play a major role in cancer? You can just write the gene name and the type of cancer. You don't have to go into too much detail. If you know of any cancer-related genes. Okay, it's fine if you don't. So one example would be the BRCA gene, BRCA, and it's a common gene that's affected, yes, in breast cancer. So after we've filtered for significant genes, we can use other sources to deduce an observable phenotype. So therefore, we'll learn about gene enrichment analysis, gene set enrichment analysis. This is used to determine pathways that are activated or deactivated in various tumors. It can help us pinpoint diseases or pathways associated with significant genes with their own statistical algorithms. We will also speak about pathway deregulation and the effect that many subtle changes can have on different conditions. We have a nice pipeline that will allow you to do this analysis in an easy and straightforward manner. Gene set enrichment and analysis will produce output pathways such as the ones on the screen associated with significant genes collected from previous steps. These pathways will give us insight into the biology of tumor development and interaction with the microenvironment. Knowing these pathways can help us deduce other relationships as well. In the next portion of this program, we will review some of the data sources and databases where you can leverage your current and future research projects. You will learn how to access different omics data from databases such as NCBI, TCGA, NCI, and COSMIC. To apply what we've learned, we will analyze data from cancer patients. We will review the most comprehensive collections of omics and clinical data found in the Cancer Genome Atlas, or TCGA. What types of cancer would you all be interested in studying? You can write a one-word answer or a sentence. Ovarian, lung, okay. Hepatic, all right. Nice. All right, nice. I'm personally interested in breast cancer due to its impact on one of my family members. Cancer Genome Atlas contains data of 12 different cancer tumor types and 6 different omics layers that pertain to that data. It has data from thousands of patients. We will learn how to navigate the Genome Data Commons portal to find multi-omics data sets with clinical information about the samples. You can then create your own projects after you develop the skills to access data with an experienced team of bioinformatics trainers. We will also cover databases like the National Cancer Institute Genomic Data Commons, an information system for storing, analyzing, and sharing genomic and clinical data from patients with cancer. The second database that we will learn about is called COSMIC, the Catalog of Somatic Mutations in Cancer. It is the world's largest and most comprehensive resource for exploring the impact of somatic mutations in human cancer. The National Cancer Institute Genomic Data Commons, GDC, has 68 projects worth of data that spans different parts of the body. As you can see on the schematic, lung, bone, marrow, and breast cancer data sets appear to be the more common cancer data sets. If a particular type of data set interests you, the Pine Biotech team can help you set up a project at a later stage in the precision oncology program. COSMIC, the Catalog of Somatic Mutations in Cancer, has more than 1,000 cancer cell lines worth of data that can be used for predictive or other types of analysis. An example shown in the bottom figure shows common variants within the classical HeLa cervical cancer cell line. Accessing related information on COSMIC can help you determine whether your observed changes within your experimental data set translates to something already known, something new, or something similar to your hypothesized results. The last part of the program will be dedicated to an independent project that you can work on with the help of our team. After learning the required skill set, you can formulate your own research question and use our user-friendly pipeline to answer that question. This project can be developed further to become an academic project for your institution or other career-level projects. Learning project planning skills is extremely useful in bioinformatics because you will probably be working on multiple projects from different scientists. Can someone briefly describe the scientific process? You can say 4 words, or you can write a sentence. You don't have to go into too much detail. Maybe the first word. Like, how would you describe the scientific process? If I want to conduct an experiment right now, what would I do? We're all biologists. I'm sure everyone knows the answer to this. Okay, yeah, observation, hypothesis. Yeah, those are nice keywords. Yeah, search for a data set. Okay. Yeah. Use a large sample size. Okay. Yep. So the process that I'm about to tell you isn't set to stone, but it's the general process. So you ask yourself, what's your research question? What rationale can you use to justify your project? What methods will you use to achieve that project? And what do you anticipate on obtaining based on your proposed pipeline or project? We will touch upon important aspects of project design and proposals looking to maximize aim of the project, that's good. Looking to maximize the value of a proposed project. Scientific projects should form a specific structure in order to ensure efficiency in time management and resource allocation. Prior literature review will give you an insight on what is out there and help you provide the foundational rationale to your proposed project. Afterwards, you can create a project proposal, which includes a rationale to your project, related methods, background on the topic of interest, and anticipated results. These steps are foundational in all types of scientific projects, such as grant writing and thesis writing. We will also go through several examples of projects to learn about successful application of data analysis tools to large omics datasets. The first example will use cell line data to classify subtypes of cancer. This project will illustrate the base scope of what you should be able to achieve after you've learned the required skill set. If you'd like to take your project to the next level, you can talk to our bioinformatics team. Can someone give me a short definition of a cell line? Or have you used cell lines before? You can say yes, or you can describe what a cell line is. Yeah, Gila, that's a famous one. Yeah. Yeah, you can give examples too. CHO cells. What do you mean by CHO cells or CHO? Like single colony cells is. Okay. Because yeah, oh yeah, okay, nice. Okay, okay. Interesting. Yeah, these are, yeah, some of these cell lines are new, or I haven't heard of one of them, the 2780, but interesting. That's awesome. So a cell line is a general term that applies to a defined population of cells. So usually they're maintained in a culture. So people use media and put different nutrients so the cells can grow constantly for certain experiments. So in this example project, we will take a data set from the publication Modeling Precision Treatment of Breast Cancer, which was featured in Genome Biology in 2013. The publication is interesting because the authors assembled a data set of about 50 different breast cancer cell lines that represent the whole spectrum of breast cancer variations. The cell lines have genomic, transcriptomic, and proteomic data, as well as drug efficacy measurements of commonly used and experimental cancer drugs. Another project example will be dedicated to liver cancer, where risk factors like obesity and viral infection can play a major role in progression and outcomes. We will take samples from the liver hepatocellular carcinoma, LIHC-TCGA project, and see how various clinical data available on those patients can be used in analysis of gene and microRNA expression. The multi-omics project uses sophisticated algorithms such as similarity network fusion, or SNF, to find correlations between different types of data. Multi-omics approaches add more insights due to the multiple levels of analysis, and you are using this more data on the same type of experiment to solve an important problem. So by the end of this course, you should achieve the following learning outcomes. You should be able to read and understand bioinformatics literature, perform basic NGS analysis, analyze data and form a hypothesis, prepare data for machine learning, cross-check signals with gene databases, know what type of data is required to answer scientific questions, create your own bioinformatics analyses, and present your results in an organized way. All of these skills are essential in bioinformatics, and some can be translated to other fields. So after achieving the learning outcomes, you would have learned some of the major best practices in the field of bioinformatics. You will be exposed to R and Python, two powerful programming languages. Bioinformaticians will need to understand how to use t-tests, regression, and other tests. Analyzing and managing data is in order to communicate important information is another best practice in bioinformatics. Pipeline development and utilization is another common practice. Ultimately, completing the precision oncology program will help you enter the field of bioinformatics, since you'll learn several best practices. At the end of the program, participants will receive a certificate of participation. As you can see on the screen here, the certificate can help you obtain a bioinformatics position in another institute, add experience to your belt, or build your CV for a graduate or undergraduate program in bioinformatics. Jobs within bioinformatics usually require experience with analyzing complex data sets, and this certification can make you stand out and give you that experience. And the link is on the bottom. We welcome you to join this program and learn about the application of bioinformatics in precision oncology. This program is geared towards citizen scientists, biologists who would like to learn important bioinformatics skills, bioinformaticians who would like to improve their existing skill set, and anyone who is interested in the field of bioinformatics with special emphasis on precision oncology. If you feel like there's a topic that we haven't mentioned within bioinformatics research in oncology, and you are interested in learning more about, please type it in the chat and we could think about integrating it for future lessons. Does anyone have any particular questions for now? You can type it in the chat.
Thank you, Clinton. We're running out of time here, so I just wanted to thank you all for joining today, and I want to point your attention to a couple of important links that Clinton and I have already shared in the chat. Here you can see the place where you can finalize your registration if you're ready to do that. So this is where you can see all of the details about the individual meetings that we have scheduled starting from March 14th. And then you will also see what topics are covered in each one of the sessions. There's an overview video. And if you're ready to complete your registration, you can just go to Get Started and see some of the options here for program registration. Now, in addition to that, we offer asynchronous learning, which is much more affordable and provides you with the same quality materials and resources that will be covered in the program itself, but without the mentorship and the interactive sessions that are included in the program. So to gain access to here, you can actually register for free. This is the link, learn.omicslogic.com. You can go inside once you have completed your registration and take a look at some of the coursework that includes getting started with bioinformatics in R and Python, introduction to data science, introductory courses to different topics and projects, including some of the projects that will be covered in this precision oncology program. Do we offer, so again, the pricing here, a lot of materials here are free, so you're welcome to get started here, and if you like it, you can explore more. There are some premium resources here that utilize some of the bioinformatics infrastructure, so those are also priced very affordably. But if you're interested in actually joining the program, we do have a process where you can apply for a scholarship. So we welcome you to, first of all, write to us at marketing@pine.bio, and you can describe there why you're seeking a scholarship, and we will send you a link to a form where you will be able to register for a scholarship and schedule an appointment with one of our team members to learn about your background and your interests. So I wanted to also let you know that this program has been completed by a number of different students, researchers, and clinicians. And here's some examples. So Olga Udartseva was a research postdoc at Roswell Park Comprehensive Cancer Center, and she has completed the training that we described today. She found that the course materials helped her gain a sense of how bioinformatics can help her, but also apply the skills that she has learned to a project, a clinical project that she was working on. We also have people that come from different backgrounds. For example, Dan Neal is a statistician at University of Florida and had to deal with a lot of research data coming from life sciences. And so he took the courses to understand where is the application of biostatistics to biomedical data. We also had a number of undergraduate students from universities like the Xavier University of Louisiana that were exposed for the first time to bioinformatics pipelines, methods of analysis, and big data. Graduate students, for example, Aaron Sugimoto, who was a graduate student at California State University in Long Beach, California, and he was pleased with the content and kind of found it interesting and applicable to his research. So we are excited to start this program. The program will start March 15th. And so we welcome you to join, and please be on the lookout for additional materials that we will be sending, including a recorded version of this webinar if you want to review it again. You're welcome to reach out to us and send any questions or requests for more information to marketing@PineBio, and we'll be able to send that over to you as well. So again, thank you everyone for joining today. We are excited to get started with this program. It's going to start very soon. And we have a number of other programs that are coming up or are ongoing. And so if you're interested in those, just feel free to reach out to us. Now, if you have any questions, we're going to stick around here for just a couple more minutes. We are already out of time. But if you do have any questions, feel free to post them in the chat. And I will stop sharing for now. And again, thank you for joining. We are looking forward to seeing you in the program.